dear fellow mates. My name is Ona Curtin, student of MSc Forestry second year, studying in College of Horticulture and Forestry. My today's title is Analysis of Phytochemical Diversity among different rhododendron arboreal genotypes in Arunachal Pradesh. This is my MSc research topic. And well, nature has a plenty of plant resources which fulfill the need of human beings, such as foods and medicines. So rhododendron tree is one of them. As we can see, that rhododendron is the flower is very red, effective color. Introduction. The scientific name is Rhododendron arboreum smiths, family belong to Acaceae, and the chromosome number bearing 26. It is the evergreen tree, and the flower bloom usually from March to April. This is a perfect timing for the flower, and so the aesthetic beauty of the flowering attract the visitors and have a rich phytochemical components, and because of that the flower has is also entitled as an state flower of Himachal Pradesh and Nagaland, and also state tree of Uttarakhand and Sikkim. It usually grown in the moist conditions and above mean sea level of 1,500 to 2,000 meters. And especially in the spring seasons, the local Himalayan regions, they used to collect the flowers and leaves and sell it into the local markets in the rupees of 50 to 100 rupees per kg. So it's significant. It defines that it has a significant commercial value also. Talking to the distributions, in whole by 1,200 species of rhododendron has been seen, and in which the Northeast Asia, Eurasia, and Western Europe and North America are there. In which China has the highest amount of rhododendron rhododendron species, which is 571 species. In India, we have 98% of rhododendron arboreum is in Himalayan region, and Arunachal is one of them, which is 90% of rhododendron are present in the Arunachal Pradesh, which are majorly found in Tawang, West Kameng, Lower Subansiri, and West Siam district of Arunachal Pradesh. Coming to the medicinal value, it has plenty of medicinal values like anti-diabetic, antioxidant, anti-cancerous, anti-inflammatory, and digestive properties as well. Apart from medicinal value, we have the flowers. They are used for the monasteries and temples, and leaves are used for the incense sticks in rituals and celebrations. Apart from this, we have value additional product also like jams, jaggery, wine, etc. Coming to the methodology, I have collected the rhododendron arboreum from Tawang and Mayagya. We collected total number of genotype 30, and the experimental design was CRD, that is completely randomized design, and I conducted my experiment in basic science and humanities labs, College of Horticulture and Forestry. The biochemical parameters which I have done was phenol, flavonoid, and phytic acid. Before coming to the estimations, let us talk about what is phytochemicals. Phytochemicals are the natural compounds which is present in the plant cell. It is important because it has so many antioxidants, anti-inflammatory compounds, and it possesses medicinal value as well. So estimations of phytochemicals. We took their samples and sorted in a different way, and then we cleaned the sample and with distilled water, and then we store in the -80 or -20 degrees Celsius. For estimations, we took 0.1 gram of samples and then we grind it with a suitable buffer, which we will be setting in the protocol. And after that, we quantify the phytochemicals using specific acids. Like in phenols, we use polyene reagents. In flavonoid, we use aluminum chloride, and for phytic acids, we use potassium tetracyanate. And after that, we put the reading in the spectrophotometer, which is run in the principle of light intensity, and then the data analysis has done. These are the results. These are my results. So as I said, I have two locations, that is Tawang and Maya. So in this graph, we can see that the Tawang has more phenolic content in the sp7 genotypes, and in Maya we have more phenolic content in SM3. By comparing these two locations, we can see that Maya has more phenolic content as compared to Tawang. Why it is like that? It may be because of the stress conditions which is present in the Maya as compared to the Tawang. That's why the phenolic content is more in the Maya side. And then the phytic acid is more in ST90 in Tawang, and in Maya it is more in SM9 genotype. When comparing both of them, we can see that Tawang has more phytic acid content as compared to Maya. This may be because of the phosphorus content in the soil of Tawang. And then we have flavonoids. In Tawang, ST1 has more number of flavonoid content, and in Maya, SM2 has more number of flavonoid content. By comparing both of them, we see that Maya has more number of phenolic content, and this may be because of the acidic soil which is present in the Maya soil. Conclusions. The finding shows that the high amount of phytochemical compounds in the plants and it is big, and that can be leads to the many pharmaceutical companies and have good potentials in utilizing in the beverage and food companies as well. The rhododendron has need to have the conservation strategies, and by this experiment, we can see that selecting the specific genotypes we can use in the breeding programs and conservation strategies.